Okay, now I want to talk about histone methyltransferase complexes. And for shorthand, I'm just going to refer to these as HMT ACEs. And I'm going to talk about three different ones. The first is the set1. Remember, set is the domain that does the methyltransferase. Set1 compass. Then I'm going to talk about Polycomb repressive complexes. And we're going to call these PRCs for short. And then we're going to talk about trithorax complexes. Okay, let's just say a little bit about this set1 compass complex here. And I want to just talk a little bit about the differences between yeast and mammals. Yeast contains the set1 protein in its complex and 7 other non-catalytic proteins. In yeast, there's just one. So now let's compare that with yeast. They also have a set1 protein in them, in the complex. They have more than 7 other non-catalytic proteins and a greater diversity. Within this group, there are some that are unique to mammals. Over here, we have just that one complex, one compass complex. In mammals, we have multiple complexes with interchangeable parts to allow for increased specificity. And that's all you need to know about the set1 compass complexes. Now I want to spend some time talking about the Polycomb repressive complexes. And we're going to come back to these in a topic that'll be later in this podcast or the next one. I want to talk about 3 proteins in this complex. The first one is this one called EZH2. EZH2 is the one that has this histone methyltransferase activity. The next one I want you to know is EED. Okay, I want you to know these 3 proteins. The first 2 are EZH2 and EED. The third one I'm going to put down here is SUZ12. So I'm going to talk about EZH2 and EED together because they have lots of similarities. They both are histone methyltransferases. EZH2 and EED are proto-oncogenes. What they will normally do is, if we draw our cell cycle here, with G1, S, and G2, and remember at this point here we have a stop that's controlled by a tumor suppressor gene. EED will block the expression of this tumor suppressor gene. And under normal circumstances, that's okay, because if there are no mutations present, we want this cell cycle to proceed. The problem comes when EED becomes an oncogene, and it's expressed all the time. And so it's always preventing this stop from occurring. And so we're always going from G1 to S. It's always inhibiting the expression of this tumor suppressor gene. So if there is a mutation that needs to be fixed or repaired, it's unable to do that. So again, EED is normally a proto-oncogene that provides an ability to get past this stop when it's time to get past this stop by turning off the tumor suppressor gene. It can be converted to an oncogene by various mechanisms, and when that happens, we blow by this stop even if there are mutations, which can lead to cancer. So I'm just going to write proto-oncogene because that's what it usually does. These will methylate several different types of lysines, including H3K27 and one of our favorites that we've grown to love and respect is H3K9. And if you remember, when H3K9 is methylated, it will turn off gene activity. Next, I want to talk about this third protein called SUZ12. It binds to heterochromatin protein 1, or HP1. By and large, its function is unclear, though we believe, because of its association with heterochromatin, that its proposed role in maintaining constitutive chromatin, heterochromatin. Interestingly, just to make sure we keep the score equal here, it may also bring into these same sites LSD1. And remember, LSD1 is a demethylase. So it's possible that SUZ12 here functions in maintaining the constitutive heterochromatin state, but when triggered to do so, can disassemble the heterochromatin. For now, that's all I'm going to say about the histone methyltransferases. The last big topic that I want to talk about is this coordination between modifying complexes. If you remember earlier on in the semester, we talked about this histone code, meaning that there should be some way to read the histones, how they were modified, in what manner they were modified, how heavily they were modified, and be able to make a prediction about the degree of expression from that area of the chromatin. So we would expect to see certain things. We would expect to see heavy methylation in some areas of the chromatin. We would expect to see heavily acetylation in some areas of the chromatin. We would expect to see a mixture of both, depending on certain factors. So let me write down what we think we would see. Areas that are actively transcribed, we would predict should have a great deal of acetylation. Remember, it's not just one acetyl group added to one lysine. There should be several areas along those histones that have been acetylated, and in multiple adjacent histones. We would also predict that we would see some adjacent methylation in these areas that are actively transcribed. To remind you, when we see this histone 3 here, we know that lysine 4 is methylated and lysine 9 is acetylated. And when that happens, we know that gene expression is turned on. This is a common trend. We've only talked about this one situation, but it's common to find areas near acetylated lysines to be methylated in actively transcribed regions. Again, as we said before, methylation is this kind of anomaly. Sometimes we see it and it indicates gene expression, sometimes it means gene repression. But in this case, it means expression. So we might predict that there should be a coordination between acetylation and methylation, between these two sets of complexes. And there is some evidence to suggest these. So let's list some evidence. We will often see multiple hat complexes working together. For instance, the histone acetylation complex called Krebs binding protein is often working at the same time that the hat 300 is working. In addition, these two are often co-immunoprecipitate with each other, indicating that they're working together and actually binding with each other. In addition, this complex, so this complex, meaning this one up here, also co-IPs, that means associates with, a known histone methyltransferase. Again, coordinating this acetylation here and the methylation here in a way to turn on gene expression. Okay, now I want to talk about sort of the reverse, and that is enzymes that work together to maintain heterochromatin state. So let me write that down this way. Complexes working together to maintain the heterochromatin state. And for this, what I want to tell you is that HDAC1, the histone deacetylases, and HDAC2 associate, both of them associate with this complex called MMSE1. MMSE1 is a histone methyltransferase, and it adds a methyl group to histone 4K20, lysine 20. And we haven't talked about this one much yet, but it is linked to repression. So what we have here, coming together, working together, are complexes that bring in these HMTases and complexes that come in to remove acetyl groups. two things that have to happen in order to maintain this heterochromatin state. It's more efficient if they come together and work together as opposed to removing the acetyl groups and then waiting for it to become methylated. If they're all right there together, they can get the job done a lot more efficiently. Okay, the last thing I want to talk about are these things called non-coding RNAs, and we'll abbreviate that with ncRNA, and how they're linked to the complexes that we've been talking about. So as we begin this discussion, I want to make sure we talk about two types of complexes. That one we've talked about a little bit before, and another that we haven't yet. The first are these Polycomb repressive complexes, the PRCs, and trithorax complexes. And when we talked about Polycomb repressive complexes before, we talked about the different proteins that were associated with these complexes: EZH2, EED, and SUZ12. One thing I want to point out to you, and I'm not sure if I mentioned this before, but PRCs will methylate H3K9s, the lysines that are linked to repression. Trithorax complexes work in an antagonistic relationship with the PRCs. They methylate histone 3 K4s. And if you remember, when we methylate a lysine 4 on the histone 3, we cause gene activation. So they're in some ways battling with each other, and they will work at the same promoter, where in some cases at the same promoter the PRCs will be active and they will cause that gene to be repressed. At other times in development, the trithorax complexes are active and they will cause those genes to be expressed. So how is the same element that brings in Polycomb repressive complexes also able to bring in the trithorax complexes? Well, I wish I could tell you that we know the answer to this and sit back and I'll tell you this great story. But we really don't. But what they think is happening is that these non-coding RNAs are involved. So how do these two complexes, the Polycomb repressive complexes and the trithorax complexes, compete with each other? Well, let's just make up a gene here. Well, we don't really have to make one up. We'll call it a Hox gene. These Hox genes are really important in establishing early developmental planes in the embryo. And so we're going to want them on early on in development and off later. So on early, off later. When they're on, these genes are on, it'll be the trithorax complex that will bind to turn them on. When we want them off, the Polycomb repressive complexes will bind to turn them off. How do we know which complexes to bind, or how does the cell know which complexes to bind? Because it's really important. Because if they don't bind at the right time, you'll end up with rather large developmental defects. Well, it turns out that there are these pieces of DNA here that we're going to call response elements. They're either Polycomb response elements, PRE, or they are trithorax response elements. So how does this change? We're not going to change the sequence, but rather we're going to change the ability of one of these to bind. And that's when our non-coding RNAs come in. It is hypothesized that there will be RNA that binds here that will attract either TRXC, the trithorax complexes, or a different RNA that could bind that might attract the Polycomb repressive complexes. These are the response elements. The response elements are the non-coding RNA, and it plays a very critical role. If the TRE element binds, it will bring in TRX complexes and turn on these Hox genes at the right time of development. If the PRE non-coding RNA binds here, it will bring in the repressive complex, the Polycomb repressive complexes, and turn off transcription. Now I should stress that there's a lot we don't know about this. about these non-coding RNAs and their role in epigenetics and turning genes on or off. So right now, this is just one of our ideas. Okay, that's all I have for these podcasts on the complexes that work to modify the histones. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.